As you can see from the title, this lecture is about heart physiology. Before we consider the functioning of the heart, I would like to remind you again the structure of the whole cardiovascular system, now with main emphasis on the heart as its center that connects the circles of circulation and provides constant blood movement. Blood movement occurs sequentially through these circles. As blood passes one circle, then the other, again and again, so that we can consider these two circles as one big circle that is formed by two loops connected in the heart, like number 8, actually. And begin from the left ventricle. Blood from this ventricle goes to the systemic circulation. First vessel is aorta, and further this aorta is branched into numerous arteries. They in turn branched into capillaries. Capillaries are gathered to the veins, and finally two large veins, inferior and superior vena cava, come back to the heart. Let's observe this process. Like blood goes through the systemic circulation to the upper and lower parts of the body, and all is finished in the right atrium, from where blood goes into the right ventricle. From the right ventricle, blood goes to the pulmonary circulation. And the first vessel is the pulmonary artery. Again, I remind you that this artery is called artery because it goes out of the heart, but it carries to the lungs the venous blood, low oxygenated, deoxygenated blood. And in order to oxygenate this blood in capillaries of the lungs, then oxygenated blood comes back to the heart through two pairs of pulmonary veins, pair from each lung correspondingly. Let's observe the passing of blood through this circle. So it comes back to the left atrium. Now it's oxygenated blood, and then it fills the left ventricle, and everything starts again and again. And the main reason of all this blood movement is the heart contractions, which produce the main propelling force for blood movement. And from the whole heart, the main impact, of course, is created by the ventricles. Their contraction pushes blood, ejects into aorta and pulmonary artery. As for atria, they have different function. This is the ventricles. The atria serve predominantly for the blood accumulation during systole. Because during systole, ventricles are in a state of contraction, and they eject blood into aorta and pulmonary artery, while the valves are closed, atrioventricular valves, and entry of blood is not possible, but blood constantly comes from veins back to the heart, and so it's necessary to have certain capacity to keep this blood that continuously comes back to the heart. And practically this is the main function of atria, to serve for blood accumulation. But also they contract, not very strongly but enough to give an additional filling of ventricles, while main filling occurs passively, and only after this additional filling is done by atria. Let's recall the heart valves, which you know from anatomy. There are two pairs of valves located in each side of the heart, which prevent backflow, movement back from ventricles to the atria during systole. These are atrioventricular valves. And backflow from aorta to pulmonary artery is prevented by semilunar valves. And blood doesn't go back to ventricles, just tries to do it and fills the semilunar valves so that they become closed. Atrioventricular valves, there are two of them, mitral for the left side, mitral or bicuspid valve, and the tricuspid valve for the right side, between right atrium and ventricle. The semilunar valves are aortic valve and pulmonary artery valve. Due to normal function of these valves, blood goes only in one direction and does not go back. There is no backflow naturally. The pumping functions of the left and right ventricles. If you are suggested to compare pumping functions of the two ventricles, in this case, many students, remembering the anatomy of the heart and remembering the great thickness of the left ventricle as compared to the right one and great size from looking outside, they immediately decide that it's easy to say the left ventricle produces higher pumping function. But we estimate pumping function not by the force of contraction. We estimate as a result of this pumping, which is volume of blood pumped each minute, for example. And let's compare now. See, the cardiac output, the blood amount that is pumped by the heart each minute, is calculated as multiplication of the stroke or systolic volume by the heart rate. From hemodynamics, I hope you deduced the conclusion that cardiac output is the amount of blood that pumped through the whole system equally. If, for example, 5 liters of blood passes through the aorta, the same volume each minute should pass through all the other vessels of systemic circulation, and similarly through pulmonary circulation, because it's the only one system. You remember that number 8. One circle, then another, then again one. So the cardiac output is equal everywhere. So the left side of equation is equal for both ventricles. Look to the right side and they're shown in green heart rate. Contractions of both ventricles occur simultaneously. There is no difference of heart rate for the right and left ventricle. And now, simply from this equation, you can conclude the stroke volume for both ventricles cannot be different. It simply cannot be different because the other components are the same. Heart rate is the same, cardiac output is the same. So let's put the main characteristics for the left ventricle. Stroke volume, the medium level is about 70 milliliters with range from 55 up to 90 milliliters, depending on the person. At rest, of course. But systolic pressure, you know values, let's say 110, 130 millimeters mercury. Then compare to the right ventricle. Right ventricle produces the same stroke volume, the same range, no difference. Just systolic pressure developed by the right ventricle is much less, 25-30 millimeters mercury. By the way, numbers coincide with the uncoated blood pressure. It's better if you remember. So, you see, pressures are different, but result of pumping, amount of blood pumped each minute is the same. The same stroke volume, the same heart rate, the same cardiac output. There is no difference for the two ventricles. Just the difference in pressure and why this difference is present. Let's recall again basic law for hemodynamics. Volume velocity, Q, is equal to the ratio of moving force, delta P, difference in pressure, with, directly proportional to the difference of pressures. And volume velocity is inversely proportional to the resistance of vessels. Or simply you may write cardiac output, because blood volume velocity measured per minute will be cardiac output, is equal to the ratio of arterial blood pressure to the total peripheral resistance. And look, we have the same cardiac output and difference in pressure. And why this difference exists? Because of the difference of the resistance of the systemic vessels and resistance of the pulmonary circle vessels. You see, ratio of pressure and resistance for both left and right ventricles is exactly the same. But on the basis of difference in pressure, you can estimate difference in resistance. Higher pressure for the left ventricle and corresponds to the higher resistance of the systemic circulation. And also The same ratio is for the pulmonary circulation. Much less pressure corresponds to much less resistance level. So we can conclude that if pressure approximately is 4 times less for the pulmonary circulation, it means that resistance of the vessels of pulmonary circulation is the same number, 4 times less approximately. It can be explained, of course, first by the shorter length of the total circle, but maybe the main reason is wider opening of the vessels, so that radius, you remember, radius is factor number 1, not compared, cannot be even compared to the length of vessels, but length also is shorter, it's clear. So higher vascular resistance of the vessels of the systemic circulation corresponds to the higher pressure developed by left ventricle. That's why this left ventricle is much thicker and greater from outside at least, but inner sizes of both ventricles are equal, and because of equal stroke volumes. And much lower vascular resistance is characteristic for pulmonary artery and the branches. Let's consider the coronary blood flow main characteristics. Facts from anatomy are arteries which supply blood to the heart. Mainly this is a left coronary artery with larger area of blood supply from this artery. And also there is right coronary artery with smaller area of blood supply. Then veins have many veins joined together form the so-called venous sinus of the heart. It's not shown in the picture. And also there is a smaller anterior coronary vein. And also they should be mentioned very little veins which are called Phibesian veins. These veins pass directly through the myocardium and open into chambers of the heart. So this adds minute little amounts of venous blood to the right side of the heart chambers and to the left side. For the right side it means venous blood comes to venous blood, but for the left side it means little addition of the venous blood into arterial blood that just has come from the lungs, fully oxygenated blood, and now it little bit added with venous blood. This is one of few reasons for not 100% saturation of blood with oxygen. It's not 100%, it's slightly less, 96, 98, or approximately like this percent of oxygenated hemoglobin. And this is one of the reasons. Main characteristics of the coronary blood supply. It's very important, the coronary blood supply for the heart functioning. First, it's important to remember that main blood supply occurs during diastole, especially in the left coronary artery, which can be compressed greatly by very powerful muscle of the left ventricle. Look, this difference for systole and diastole for the left coronary artery. During systole it becomes approximately 2 to 3 times less as compared to the diastolic period. Then, right coronary artery has not such a great difference and just slightly has decreased blood supply as compared to diastole, because the right ventricle, you know already, contracts with much less force. Then, important numbers, easy to remember, 5 and 0.5. High level of blood supply can be shown by relative comparison. Just imagine if all tissues get equal blood supply and each organ gets oxygen amount and blood amount proportionally to the weight of the organ. So the heart has 5% of cardiac output blood supply, but heart itself as an organ makes just 0.5% of the body mass as a whole. So, according to these numbers, you can see that compared to average, it's 10 times higher blood supply than equal to every organ. And it's easy to understand. First, heart has no great rest. Of course, it has a little rest during the cardiac cycle, but generally it operates regularly, constantly. But also there are other reasons for high level of blood supply, which we consider later again. Then, capillary network of the heart is extremely dense. This high density can be estimated by counting of number of capillaries, as usually it's done, number of capillaries per millimeter of tissue cross-section. And you may see more than 2,000 capillaries per each square millimeter. It's a great amount, and it's close to the brain blood supply actually. Then, high sensitivity to the lack of oxygen and easy effective dilation of arteries in response to oxygen deficiency. It looks very logical that oxygen deficiency produces dilation of arteries, but as we will see later when we study the regulation of vessels and arterial blood pressure, you will see that most of arteries respond by dilation to the other factor, excess of CO2, and much less sensitive are to the oxygen deficiency. But heart is greatly sensitive to the oxygen deficiency. Again, it shows how important is oxygen for the heart. And when oxygen deficiency occurs, coronary vessels dilate, and this increases the coronary blood supply. And maximal increase can reach level about 3, even 4 times more than at rest. And we can calculate, using this percentage given about 5% of cardiac output, we may calculate the absolute value of resting blood supply to the heart. And also we can calculate correspondingly the maximal. So 5% of classical 5 liters makes 250 milliliters, or a quarter of liter each minute at rest. And if this quarter is multiplied by 4 times maximally possible, you can see that in case of intensive physical activity, the heart may get blood supply up to 1 liter each minute to give necessary oxygen for activity of the heart. Characteristics of the metabolism of the heart muscle. It's also very important. First feature is easy. It's only aerobic, mainly aerobic oxidation. Let's recall the muscles can have aerobic and anaerobic metabolism, but the heart uses mostly aerobic. This is one additional factor for high requirement in oxygen. But number one factor, perhaps intensity of work, of course, but the types of substrates, because it also depends on the substrate for oxidation, how much oxygen is required. Different substrates require different amount of oxygen. And heart, it also can be mentioned as a separate feature of metabolism, that it can metabolize, can oxidate different substrates. Not only one, but practically all possible, all available. For example, the skeletal muscle mostly use glucose. The brain uses only glucose, and so on. Heart can use everything. But at rest, the first place is occupied among all the substrates, occupied by free fatty acids, more than 70%. And perhaps this is the main reason why one of the main, at least, reasons why heart has such great requirement in oxygen. You know, fats can give a lot of energy, because each gram of fat gives 9 kilocalories. Nothing else can give it, because both proteins and carbohydrates give only 4 kilocalories. But to oxidate fats, it takes a lot of oxygen. The fat molecules themselves do have not so much oxygen inserted. So to produce the same products like CO2 and H2O, water, the same as carbohydrates may give, we need to add much more oxygen molecules. So oxygen requirement is determined by, perhaps mostly by the substrate used for oxidation, number one. Second place is occupied by glucose. As I said, all the substrates can be used. The third place by lactic acid, lactate. And there are some other with very little percentage, not mentioned on this page, but it can be oxidated pyruvic acid. Heart may oxidate, of course, not big amount, not big percentage of the ketone bodies and other not fully oxidated products and bring them to the full oxidation. But this amount is negligible in percent. So these three are main substrates for resting state. For a case of physical activity, exercise, when heart should work stronger, the position of these three substrates is changed in such a way that lactate occupies the first place, up to 60%, and shifts the other substrates, such as fatty acids, to the second place, and glucose correspondingly to the third place moves. It's very good that we can use lactate for the heart, because when we perform strong exercises, the skeletal muscles produce additional more and more lactate. At first they manage to oxidate fully glucose, but gradually it stops in the pyruvic acid stage, when pyruvic acid quickly becomes converted to lactate and it becomes accumulated, thus creating the lactic acidosis, which decreases the ability of muscle to contract further and produces muscle fatigue. But the heart muscle can take this lactate and oxidate fully, and thus decreases the lactate amount, decreases the degree of lactic acidosis. That's very good. And we can prove the high level of oxygen requirement by calculation of the important index. This index is called oxygen utilization quotient. It's like coefficient, but usually called quotient. This is the part, actually percentage of oxygen that is utilized by tissue from the arterial blood. Content of oxygen in the arterial blood is taken for 100%, and each tissue takes different percentage of this oxygen, and never 100, never. Basically, the whole body, taken as a whole, at rest takes about a quarter of this oxygen, about 25% on average. But look at these values. Actually, the skeletal, for comparison, you can see here skeletal muscle and cardiac both. At rest, skeletal muscle uses about 30–40% of oxygen from the arterial blood. And at exercise, it can be increased from 40 up to 50 and 60 maximally. But cardiac muscle, even at rest, has oxygen utilization minimally 60%. What is maximum for the body as a whole and for skeletal muscle separately. So when activity increases, this percentage becomes even higher, up to 80%. So you may imagine that venous blood that goes out from the heart is different from the every mixed venous blood. It contains much less oxygen that the other organs may have in venous blood because of much greater percentage of oxygen utilization. The arterial blood is supplied one and the same, with the same content of oxygen to all the tissues, but venous blood that passes from the organs may contain more or less oxygen depending on how much oxygen is taken by the tissue. And cardiac muscle takes more than practically all the others. So percentage even at rest, 60–70, oxygen utilization quotient. Now we start to consider the heart muscle tissue itself. And first thing we should mention that it consists of the two types of cell. Contractile muscle cells. Of course, they compose most of the heart. These cells are the main cardiac cells, and they are present in both atria and ventricles. There is slight difference between atria and ventricles, atrial and ventricle muscle cells, but it's little difference, negligible. Basic differences between contractile cells and the other that we are going to consider later. The main cardiac cells contract when they become excited by action potential. But this action potential should be sent to these cells. They cannot produce the action potential themselves, they cannot become excited themselves. They only follow the excitation that is sent to them. Then they contain myofibrils, of course, with actin and myosin, because it's necessary for effective contraction. then they are connected by the gap junctions, which makes a big difference from skeletal muscle cells, which do not have any gap junctions, and each individual cell should get stimulation from nerve ending individually. And if one cell is surrounded by excited cells and this cell does not get impulses, it will not contract, will not become excited. For the heart, we cannot excite just a part of cells. Among contractile muscle cells, it's enough to send impulses to the part of them, and all the rest, their neighbors, will get impulse from each other. So functionally, as these cells are, of course, separate cells, they functionally operate as a single one. This is called functional syncytium. Syn connection, and cytos is cell, so syncytium is connection of many cells and functioning as the single whole one. This is important for the heart, and this is the basis for all or nothing law, if you remember. The resting potential of these cells is similar to the resting potential of the skeletal muscle cells. Average about -90, sometimes -55 or so. And entirely different, very different type of cells are conductive system cells. Of course, these cells are less numerous, and they form the conductive system of the heart with its nodes and bundles and fibers. These cells are able to automatic depolarization and excitation. So they generate impulses that control the heart contractions. Exactly these cells send impulses to contractile muscle cells and drive them to contraction. These cells are not good in contractions and they don't need great number of myofibrils. They contain a few myofibrils. They do not contain sarcoplasmic reticulum. It's not their function to contract. And their resting potential is closer to potential of the nervous cell. So their function is almost also close to functions of nervous cell. And what is very important, this potential is not stable. Then usually excitable cells, if they have resting potential and nothing stimulates this cell, they remain at their resting state, the same level of potential. But the conductive system cells do not keep this potential stably at the same -60. After each time when they reach this -60 value approximately, they gradually start to depolarize. This potential is not kept at a steady level. It tends to go to less and less negative values. On the figure it goes upward, and this is depolarization. So let's start from the cells that are reason of heart contraction, the conductive system cells. But before, let's recall the structure of the whole conductive system. Conductive system consists of nodes and bundles with fibers. First thing to remember also, it's necessary to keep in memory that we have separate myocardium of the atria and separate myocardium of ventricles. Everywhere through atria, conduction is easy from cell to cell. Also in ventricles, conduction easily goes from cell to cell. But from atria to ventricles, conduction cannot occur from cell of contractile myocardiocytes to cell, because there is separation by fibrous tissue, like isolation tissue, isolation things we use in electricity. Annulus fibrosis, this is fibrous ring that surrounds valvular openings between atria and ventricles and entirely stops contact between atria and ventricles. So it's important for understanding why excitation doesn't come straightly from atria from all the sides to the ventricles. No, excitation from atria can pass to the ventricles only through the atrioventricular bundle. So this bundle passes through this fibrous ring, annulus fibrosis, and then the bundle is divided into right and left bundles and goes further to the myocardium of ventricles. And exactly here in atrioventricular node, that is the beginning of atrioventricular bundle, occurs the delay of conduction of excitation. We'll come back to this to consider again, but
This is important for given time interval enough to finish the atrial contraction, and just then excitation and slow contraction of the ventricles can begin. This time is slightly over 0.1 second, and it gives opportunity for the ventricles to start their contraction only after end of atrial contraction, atrioventricular delay. Let's recall the full conductive system of the heart, its structure, because exactly according to this structure and direction of bundles and pathways, the excitation of the heart goes. So the first node of the conductive system of the heart is the Sinoatrial Node. It comes from two words, sinus. Sinus is a dilated portion of the atria nearby the superior vena cava. So sinus, and atrium, so sinoatrial node. But you may simply name it sinus node, it's shorter and easier. Sinus node begins everything, and exactly here excitation appears first. Then excitation is conducted from sinus node cells through the conductive system cells arranged into fibers, bundles. These fibers are 3 to 4, and one goes to the left atrium and few goes along the walls of the right atrium, and they all finally come to the atrioventricular node, the next node of the conductive system. And conduction between cells of the conductive system through these bundles of conductive system fibers goes with velocity approximately 1 meter per second. Taking into account that distance between two nodes is very, very short, the excitation of atrioventricular node could occur very soon, and in this case, if conduction goes with the same velocity, there will be great probability that ventricles start to contract just when atria are in the stage of their contraction somewhere before reaching the middle. So if atria contract simultaneously with ventricles, or ventricles contract simultaneously, you can suppose which structure will win, which structure develops higher pressure. So atrial contraction will be entirely useless, and nothing will be pumped into ventricles, because ventricles will overcome, of course, the pressure created by atria. So we really need this delay, which occurs exactly in the atrioventricular node, where the velocity of conduction suddenly falls as low as few centimeters. Exactly here in the atrioventricular node, conduction goes with velocity of few centimeters per second, in particular 2 to 5 centimeters per second. It's great decrease of velocity of conduction, and exactly this provides atrioventricular delay. Somehow the contacts between cells are arranged in such a way that conduction cannot be fast. And so it helps to produce sequence. Atria contract first, ventricles later. Then, after excitation of atrioventricular node, the atrioventricular bundle goes through the atrioventricular septum and passes to interventricular septum. So, atrioventricular node itself is located very close to ventricles, but still in atria, and bundle should pass down to ventricle. And then, for a short distance, approximately about 1 centimeter plus minus, it goes down, and then it branches into two branches. This is called bundle of His, historically. This bundle of His, as you remember from anatomy, is branched into right and left legs or branches. And they go beneath the endocardium down to the apex of the heart. And on their way, of course, they give numerous branches, and these branches are called already Purkinje fibers. These legs and branching are formed by Purkinje fibers. And velocity of conduction again becomes faster, and really it becomes faster than even in atria. And it gradually becomes faster and faster progressively, and reaches maximal level in final fibers up to 4 meters per second. This great velocity of conduction of impulses through the ventricles provides very
quick involvement of ventricles into contraction, so that within a very little time all ventricles become involved into contraction. It provides great simultaneous contraction of ventricles. Of course, there is little negligible difference, as you can see from the pathway of conductive system. The interventricular septum starts to contract earlier, and apex starts to contract earlier than upper portions of the ventricles. So it creates direction of blood ejection upward to the vessels, aorta and pulmonary artery. And this is important. But this difference is very little, and generally within shortest time, less than 1/10 of a second, few hundredths of a second, the whole ventricles become involved into excitation and correspondingly become contracted. Now we start to consider the most complex part, action potentials. And two different types of potentials exist in the heart. Of course, there is a little difference between contractile muscle cells, potentials between atria and ventricles. But this mostly refers to duration, which is shorter for potential in ventricles, but basically it's the same potential. But potential of the cells of the conductive system is very different from potential of the cells of contractile main myocardium. And it's nice, always widespread mistake, the mixed two potentials during examination, and students always represent, not almost always represent opposite potentials, opposite to what was asked. Try to pay attention and to understand, and it becomes much easier to remember which potentials is for which cell. Action potentials of pacemaker cell, we'll consider what is pacemaker later. It's the cells of the conductive system, and these potentials are generated automatically. So, if you remember that there is one phase which goes automatically, specific phase, you'll easily remember how this potential looks like, and will never mix with other potentials. So each potential generated by the cells of the conductive system results in contraction of the heart. So rate of impulses should be equal to the rate of the heart contractions. These rhythmical generational potentials control the heart contractions, and it occurs from the sinus node. This is a general look of the potential of the conductive system cells, and basically for sinus node. And notice potential level minus 60, that corresponds to resting level, is not maintained for a long time. It's just small points repeatedly appearing in a regular changing of potential, and after reaching minus 60, potential starts to go rather slowly upward and upward, which is depolarization. This depolarization reaches the threshold and produces generation of excitation with quick depolarization, rather quick relatively, then repolarization. When it comes back to minus 60, again everything is repeated. And exactly this slow diastolic depolarization phase is automatic and base for generation of impulses without any external influences by sinus node and other cells of conductive system. You see, when threshold minus 40 millivolt is reached, it results in opening of many channels that produce depolarization, and potential is generated. Actually, all the excitable cells do the same when they reach the threshold, but most of cells are not able to reach the threshold without any stimulation. They need external action potentials that bring their potential to threshold. But these cells slowly, gradually reach the threshold themselves. So this is spontaneous, without external reasons, internal depolarization. And it's rather slow. You see, fast depolarization go upward almost vertically or close to it. And this depolarization goes under angle of maybe 30 degrees, and so it takes time to reach that threshold. But when the threshold is reached, potential develops. And totally, this, as you can see, consists of these three portions. this slow reaching the threshold, then upward depolarization and downward repolarization. And then all these three phases are repeated again and again. Now let's consider how this potential is generated, which ions are involved into this, which channels. So the mechanism of the slow depolarization, the diastolic depolarization phase. It's called diastolic depolarization because exactly it occurs during diastole. When cell gradually reaches the threshold, it's still not excitation. It's still approaching the excitation. And at this moment, the heart is in the state of diastolic relaxation. But when action potential is generated, it starts to be conducted down the conductive system, and finally it involves into excitation and produces contraction of the heart muscle. So this is cell. Let's say that this is the sinus node cell. As all the excitable cells, it has higher potassium concentration inside, and usually they have rather good permeability for potassium that allows potassium to go down the gradient out. When potassium goes out from the cell, it creates negativity predominating inside. And thus, usually we form the resting membrane potential. And here this resting potential also is achieved at the level minus 60 millivolt. But this resting potential for these cells is unstable. It cannot be maintained constantly at minus 60, and it slowly tends to decrease and decrease, which is depolarization. So let's make the figure. Minus 60 is the resting level. 0 is necessary to mention, plus 10, which will be peak point, and minus 40, which is the threshold, minus 40 millivolt. And so, slow depolarization phase occurs due to participation of the three ions that take part in this. All three ions' permeabilities become changed. Due to permeability change for sodium and calcium, which increases. Actually, it's through one and the same type of channels which are permeable for both ions, sodium and calcium. These channels gradually appear to be open, more and more channels. Not quickly, not many, but enough to allow positive ions to enter the cell. And these ions go into the cell. At the same time, somehow, potassium permeability becomes decreased. Channels for potassium start to close, which decreases opportunity for potassium to go out. And so you can see that not quickly, slowly, all three ions, all positive, become accumulated inside the cell, which brings its initial negative charge to progressively less and less and less negative. And this looks like the slow diastolic depolarization. As I already said, for regular classical heart rate with sinus node, it makes the angle about 30 degrees approximately. And when finally it reaches the threshold, here we have generation of depolarization much faster. So again, three ions, sodium, calcium, and potassium. And all these changes of permeability contribute to depolarization. Sodium and calcium extracellular ions enter the cell, and the permeability is higher, allowing them to enter. And potassium, intracellular ion that usually goes out, now cannot go out as much as before, and so it remains in the cell, and its positive charge also contributes to depolarization. So if you remember that all the direction of changes of permeability is contributing to depolarization, you easily may predict which change should occur. For sodium and calcium, higher permeability, for potassium, lower. Then we reach the threshold, and slow diastolic depolarization phase is ended, and next phase starts. Much more channels become open for sodium and calcium, and rather quick depolarization occurs. Just here, we don't have the sharp peak. The peak is rounded. So calcium and sodium entry produce depolarization phase simply, not slow but not fast. We don't call it fast. It's faster clearly, but name is not included. And then these channels at the upper point become closed for calcium and sodium, and potassium channels become open, which allows potassium to go out of the cell and restore back negative charge in the cell. So this is a phase repolarization, when potassium removes positivity and restores negative -60 charge. But by this time, when we reach again this potential, permeability for potassium again starts to decrease, and for sodium and calcium increase. And we gradually have again the slow diastolic depolarization phase, and everything becomes repeated and repeated and repeated, and it goes through the whole life of a person. And normally each impulse goes down to the muscle of heart and produces contraction, so that action potential frequency equals to the heart rate. Now let's consider about what is the automaticity gradient. Ability of the conductive system cells to generate impulses is called automaticity. But automaticity is different for different cells, and exactly this difference is called gradient of automaticity. And this gradient goes exactly from sinus node downward with gradual less and less automaticity. How we can estimate automaticity? By velocity of the slow diastolic depolarization. Actually, if this velocity is changed, the duration of impulse as a whole, potential as a whole, is changed as well. And if it goes slower, so the duration of each cycle becomes longer. And with longer duration of each cycle, the heart rate, impulses rate of course first, then a heart rate correspondingly becomes less. So the gradient of automaticity means that the cells of the first node, sinus node, have fastest velocity of the slow diastolic depolarization. It's still slow, but among other cells, it's fastest. If we go down to AV node, atrioventricular node, it also has the ability to generate impulses automatically. It also has slow diastolic phase of depolarization phase, but it goes slower. And as we go far and far from sinus node along the way of the conductive system, this velocity of depolarization becomes slower and slower, and this is gradient. And as a result, we can understand why sinus node is the main giving order to the heart, which is we call pacemaker, making the pace of the heart, because it generates impulses first. Now we have 4 cases of the similar figures for different parts of the heart. First, there will be slow diastolic depolarization and potential for the sinus node. Beneath, there will be AV node, and AV node, as you can see, develops the slow diastolic depolarization slower, and corresponding threshold is reached later. Then, even slower it goes for bundle of His, and the slowest for Purkinje fibers. So you may understand that while the AV node and all the rest lower located parts and cells of the conductive system produce their slow diastolic depolarization, they have not enough time to finish this and to reach the threshold, because sinus node has already generated impulse and very quickly sent it down. And even taken into account the AV delay that occurs in the atrioventricular node, anyway, the impulse comes earlier, before AV node can reach itself, its threshold level -40. So impulse becomes generated instantly in the AV node due to stimulation by action potential conducted from the sinus node. So AV node now follows the rhythm given by the sinus node. And the other parts of the heart clearly do the same, because after AV node, delay is not present anymore and conduction goes much faster, almost instantly downward, just with little decline. And so all the rest parts of the conductive system become excited by impulse that came from the sinus node. And sinus node is the pacemaker of the heart. So it drives the rhythm of the heart contractions because it generates impulses earlier. But now, why all the cells are able to generate it if we don't use it? What's the meaning? What is the significance of this? I hope you understand that. Under normal conditions, their activity is not necessary, their automatic generation of impulses. But it's produced very good help in case of any problem with conduction of impulses from sinus node. And it happens in pathology. In many cases, it can happen. It's abnormal, but it happens when conduction somehow is stopped. Mostly it happens between the sinus and AV node somehow, and it's called atrioventricular block or blockade. But blockade can happen in lower levels. It may be due to many reasons. For example, the vagus nerve high activity can produce sometimes very big slow down of the conduction, and excessive stimulation of vagus may produce even stop of conduction. Some other reasons, mechanical reasons, that normal blood supply sometimes different and clear reason may stop conduction of impulse from the sinus node down. Most often this occurs between the sinus and AV node. So now sinus node cannot reach the AV node and activate it. Atrioventricular block stops activation of AV node by sinus node, and now atrioventricular node can reach the threshold itself and produce its action potential. But you see that as slow diastolic depolarization takes longer time, the frequency of impulses and corresponding to the heart rate produced by AV node activation will be less. If we take the sinus node average rate 75 per minute with 60 to 90 variability, the average value of contractions, impulses and correspondingly contractions produced by AV node activity is 40 to 60. Then less often it happens, but also may happen if the block occurs lower than AV node, somewhere between atrioventricular node and bundle of His. In this case, bundle of His will drive the ventricular contraction, but it can take longer time to produce impulses, and impulses rate will become 30 to 40. And when Purkinje fibers become the pacemaker of the heart, when bundle of His cannot conduct impulses down to Purkinje fibers, there will be rate of impulses even lower, 20 to 30 impulses. With such a rate of impulses, the heart contractions with corresponding rate will become not enough to maintain proper blood supply to brain, to maintain normal consciousness, so person may become unconscious for a moment. And also you should understand such a thing that according to these figures, it looks like immediately after block development, the impulses should be generated by next by latent pacemaker. But really it doesn't happen like this, because for a long time under normal conditions the activity of AV node and all the down located lower areas, parts of the conductive system are suppressed. This activity is suppressed, and to restore it and to return back to its own automatic generation of impulses, they need some time, and this time usually is between 5 to 20 seconds at least. Of course, it looks like very short time, 20 seconds, and not to say about 5. But even 5 seconds means more than 5, 6, 7, 8 of missed heart contractions. So it means the heart stops. There is no blood supply to aorta, and mainly what will react first? The brain. Without blood supply, brain will stop consciousness and person simply drops down. So it's dangerous if it happens abruptly, suddenly. In these cases, people should use the artificial pacemaker when they have this great danger of stop of conduction suddenly. So now I hope you understand the automaticity gradient, that all the parts of conductive system are able to do it, but naturally the activity of lower portions is suppressed by sinus node, which is natural pacemaker of the heart. The other parts can become pacemaker, so we say they are latent pacemaker, but under normal conditions their activity is suppressed and they don't generate automatically impulses and simply become excited by impulses that come from the sinus node. Now we switch to the contractile muscle cells, which form the most of the heart. Contractile muscle cells are, as you remember, prolonged and branched cell with central single nucleus. What is important to notice here, they have connection between cells that are intercalated discs, and these discs are actually gap junctions, which allow passage of electrolytes and therefore easy conduction of depolarization from cell to cell. Again, I hope you remember that this easy conduction produces the functionally operation of the heart muscle as a single whole. Of course, again, atrial syncytium separately and ventricular syncytium separately. But within ventricles, for example, all the cells are connected to each other, and conductive system Purkinje fibers come not to each individual cell, but to part of them. And the rest, their neighbors, simply get impulses from stimulated cells easily. And heart always participates entirely in contraction. Normal heart cannot be excited partially and contracted partially. All the cells participate in contraction. This is the basis for all or nothing law of the heart from excitable tissues law. Now we come again to the potential, very complex potential of the cardiac muscle cells. And it consists not of the three phases, and there is nothing spontaneous and nothing slow, or no, slow is present. Here, level of resting potential is stable and approximately minus 90 millivolts, similarly to the skeletal muscle cells. And if cardiac muscle cells do not receive impulses, their level of potential, resting potential minus 90, will remain constant, unchanged. There is no any tendency for depolarization automatically. Before consideration of the action potential of a muscle cell, it makes much easier understanding if we compare to the skeletal muscle cell action potential. And skeletal muscle action potential is very, very simple, one of the most primitive. Just quick or simply fast depolarization produced by opening of the fast sodium channels, which goes upward almost vertically and abruptly stops because of inactivation. Gates of sodium channels are closed. Just at this moment, opening of potassium channels begins, and opening of potassium provides opportunity for potassium to go out from cell and produce restoration of negativity minus 90. So this potential consists of only two phases, depolarization and repolarization. This is skeletal muscle cell. Note that the restoration goes directly to the resting level, no hyperpolarization. And so you see two types of channels are present, like in neurons, fast sodium channels and potassium channels. The same channels are present in the cardiac muscle cells, but additionally there are other channels. But it begins similarly. Potential begins from opening of the fast sodium channels. So sodium enters quickly, and again quickly the gates for inactivation are closed. Then there begins slight increase of potassium. The potassium permeability becomes, in the beginning of potential, less than resting level, so decrease occurs. And now gradually it starts to become restored. And first potassium ions start to move outward, beginning the phase of repolarization, which is initial repolarization. But then, finally, other channels start to be open. These other channels are slow calcium channels, which make big difference. These channels are not present in skeletal muscle cells, but here they are present. They are called slow because they open slowly and close extremely slowly. So you see, from the beginning of potential, the calcium channel start to be open only approximately at this moment. And instead of theoretically restoration quick like in skeletal muscle, the opening of calcium slow channel delays this repolarization, delays many times. You see, while skeletal muscle potential lasts very short time, this potential is very long, and approximately it takes 0.3 second. You see, tenth portion of second. Compare the sodium channels open few 10,000th portion of the second. Here a few tenths, so difference is 1,000 time, as you can calculate easily. And what happens here? These slow calcium channels allow entry of calcium down the gradient into the cell, but potential stays at the same, almost the same level because of balancing of positive ions entry by potassium going out. And these two processes are almost balanced, and time goes and goes, and there is no change of potential. It remains above zero, so sodium channels stay in their inactivated state. No repolarization, no restoration of Gates position. And this takes a long time. During all this time, calcium channels are open. And finally, when calcium channels close, after this almost 3/10 of a second, the potassium, which still goes through channels outside, rather quickly restores potential back to resting state. So you see many phases. According to colors, even you can see there are at least 4 phases, but they are numbered, not logical like 1, 2, 3, 4, but different. It's accepted the standard way of numbering, and you should be familiar with this because it's used everywhere in pharmacology in different points. So you should know that first fast depolarization is numbered as fast phase 0. Then little initial repolarization is named number 1 for potassium. This is initial fast repolarization. Then repolarization is slowed down greatly, and the phase number 2 is called slow repolarization or plateau phase. Why plateau? It's easy because it looks really like a mountain with very flat apex, flat surface upward. So plateau phase or slow repolarization. And then repolarization in phase 3 becomes again fast final repolarization. And number 4 is placed for the resting membrane potential. So it's established way for numbering of these phases, so you should be familiar. And now, this is the permeability of calcium channels. You see, it increases slowly, and really when it becomes really increased, the phase number 2 starts, when repolarization is slowed down. And when permeability for potassium starts to be decreased, it means channels become closed and closed, the plateau phase is ended. So permeability for calcium approximately is elevated during the plateau phase, and this is the reason of plateau phase. And you see these slow channels and usually are open for very, very long time, few tenths of a second. The whole potential lasts for 300 milliseconds or 0.3, and big portion of this, bigger portion, is taken 90% at least by this plateau phase. And here we are going to place the excitability. And again, to make it simpler, let's start from simple excitability of the skeletal muscle cell. Lower line will be 0 and upper line will be full excitability, initial. It can be taken for 1 or for 100%. So when sodium channels become open, they cannot respond to new depolarization by opening, because they should be closed first. They should restore the initial position of gates. And so from the moment of sodium channels opening, the excitability becomes exactly zero, drops to zero immediately. Then it remains at zero level until repolarization allows to restore position of gates and put everything as it was before. And now channels, if all the channels are restored, the full restoration of excitability. But excitability is restored within the short period from zero level up to full restoration of all the channels. As a result, you can see it corresponds, this figure of the excitability is very short, corresponding to the short duration of the action potential of the skeletal muscle. And absolute refractory period, when excitability is entirely absent, zero level, is very short. And what is the significance of all this? First, let's compare with the cardiac muscle. At first, it goes similarly. Sodium channels are open and they cannot respond to new stimulation from the opening moment. So, immediately the excitability drops to zero. But then, sodium channels remain inactivated until the phase 3 beginning, because there is no way to restore position of channels. Membrane has opposite charges to normal. And inactivation gates of the sodium channels remain closed, while the activation gates remain open. And this channel cannot respond to any stimulation. And as the phase number 2, plateau phase, goes and goes, last and last, the excitability again remains at the zero level until the beginning of phase 3. So it produces very long absolute refractory period. And this refractory period includes phases 0, when sodium channels are open, phase 1 and phase 2, when sodium channels are inactivated. And only during the phase 3, restoration of negativity inside produces restoration of sodium channels, and ability to become excited becomes gradually restored quickly. And actually here we can produce slight hyper excitability in the end, but here I didn't show it. But then excitability finally is restored. Long absolute refractory period. And what's the use of this now? Let's compare again cardiac muscle and skeletal muscle. Here, about phase duration, I said that 300 milliseconds for the potential as a whole, and 90%, 270 milliseconds approximately, is taken by the absolute refractory period, within which the cardiac muscle cannot respond to induced stimulation. We'll simply not notice this. So compare. Here, this is a moment when red arrow appears. This is stimulation of muscle cell, and green line shows the mechanical process of contraction. I hope you remember 3 phases. Latent period, no contraction, because generation and conduction of action potential takes time. And finally, it should release calcium, and only then contraction begins, shortening of the muscle. And this shortening is shown by ascending portion of line. Descending portion of the green line shows the relaxation of muscle. Contraction and relaxation figure. And all this time, excitability is already normal, because action potential is over, refractory period is over. So any stimulation during this normal excitability time produces summation, produces generation of new impulse. This new impulse will open additional amount of channels for calcium in reticulum. More calcium will be released, and contraction will become stronger. It becomes summated. And the type of this summation depends on timing of the next impulse coming. Here is the border between contraction and relaxation. If contraction is not followed still by relaxation and new impulse comes, so this contraction without any interruption will become gradually stronger and stronger. This is smooth tetanic summation, if you remember. So without any relaxation, muscle contracts and then contracts even stronger and stronger. But if next impulse comes after relaxation beginning, there will be attempt to relax, and then new contraction starts. So there will not smooth movement, it will be serrate tetanus, which is not physiological, it's mostly laboratory phenomenon. Our normal muscle contractions are smooth, we gradually increase the force of contraction, gradually decrease. But imagine if something like this happens to the cardiac muscle. Left ventricle, just imagine, contracts, releases the systolic volume into aorta, and without relaxation starts to contract stronger and stronger and stronger due to stimulation by impulses. In this case, there is nothing which can help to maintain blood movement because contraction of the heart will be useless. It's useful when it is filled with blood, then contracts. Without new filling, this contraction is entirely not necessary. So it's equal to simply relaxation of the heart and stop. In both cases, heart stops. In systole or diastole, no difference. So the pumping function is entirely stopped, failed. And the heart really never comes to such type of summation like smooth tetanus. And why? Because the long absolute refractory period. You see how convenient is it? This calcium that enters contributes to the force of contraction because calcium binds to troponin and participates in interaction between actin and myosin filaments. And at the same time, this calcium keeps membrane depolarized, prevents the repolarization, and prolongs the absolute refractory period so that the heart muscle, even if it gets unnecessary impulses from somewhere, pathological impulses, it doesn't respond at all because there is no way to generate new impulse. So heart muscle doesn't start any summational contraction like a smooth tetanus, for example. But still, the additional heart contraction is possible pathologically if next impulse comes within the period that goes after the potential. Let's make a figure, also green line, for the contraction of the heart. Contraction begins when calcium appears. So the beginning will coincide approximately with increase of permeability for potassium. And again, ascending line shows systolic contraction, and relaxation that occurs during diastole is a descending portion. You see, systole almost entirely covers the potential, and almost entirely systole is accompanied by long absolute refractory period. So when ventricles contract during systole, they do not respond to any additional impulses that could produce tetanus or summation. Fortunately, we are protected from this. But later, when potential and absolute refractory period is over, during all this period, we cannot stimulate the heart. But here, when this green line goes, normal excitability is restored. Then, if unnecessary impulse will come, it will produce additional heart contraction. And this is called extrasystole. Extrasystole may come when relaxation is not finished entirely, and sometimes it may happen even after full relaxation, but earlier than normal impulse will produce next regular heart contraction. So this is extrasystole. Then let's again follow the phases and ion mechanisms of the 4 phases. Phase 0, upgoing. This is fast depolarization from -90 up to +20 millivolt. What is the basis for this phase? Opening of the fast sodium channels and sodium influx into the cell, phase 0. Also, at this moment, potassium permeability decreases as compared to resting state. And in the end of this phase, just little beginning of permeability for calcium increase occurs. Just a little, it starts. Then, phase 1, initial fast repolarization. This small portion, phase number 1. And it's produced by, first, why previous phase stops. Exactly, stop of previous phase is achieved by inactivation of fast sodium channels, so that line doesn't go upward anymore. But exactly this decrease of the depolarization degree, this attempt to become repolarized, occurs due to elevation of the potassium permeability. It starts to be higher after drop down in the fast depolarization time. Then phase 2, slow repolarization or plateau phase. Look, this one, phase when potential almost doesn't change for a long time. This phase is created by two opposite movements of the ions. First, calcium permeability now becomes increased due to opening of the slow calcium channels, and calcium goes into the cell. But simultaneously, potassium goes outside continuously, and therefore influx of calcium approximately is equal to potassium efflux. Asterisk here means that actually number of ions should not be equal, because calcium brings two positive charges, so number of potassium ions, absolute number, should be twice higher to produce full balance. But generally we consider that on the basis of charges, these two processes are equal. And as a result, repolarization is delayed, and we don't have this decline of the figure. And phase 3, final fast repolarization, occurs due to this. Phase number 3 occurs due to closure of calcium channels. They stayed very long time open, but finally their closure comes. And potassium, which still is able to go through open channels for potassium easily brings repolarization. When nothing interferes to do it, no more entry of calcium. And finally, phase number 4 simply means the restoration and resting membrane potential. I hope with this lined explanation of each phase, it becomes easier to understand and to memorize. Now, let's recall something that is characteristic also for the skeletal muscle, the excitation, contraction, coupling. Joining of the two processes, electrical process of excitation and mechanical process of contraction. What is the intermediate factor between these two? This is calcium ions. Connection between electrical process of depolarization, action potential traveling along the membrane of fiber, heart muscle fiber, and mechanical process of shortening occurs due to electrical processes, activation of calcium entry into the cell, entry into the cytoplasm, and then calcium ions provide mechanical shortening activation. Here you see the model of the piece of membrane of heart muscle cell with T-tubule. T-tubules are very well developed in the heart muscle, much larger, maybe 5 times larger than in skeletal muscle. And calcium source for contraction is not only reticulum, which is less developed in the heart as compared to the skeletal muscle, but T-tubules are much more developed, and through T-tubules, calcium channels, calcium comes from the extracellular fluid. Extracellular fluid is one source of calcium for contraction, and sarcoplasmic reticulum is the source number 2, not the first one. But both participate. So let's consider how it goes. Calcium is present in both sides, outside and in the reticulum. First, to excite the cell, it's necessary to send impulse from conductive system cells. And when conductive system sends this impulse, the membrane of the cardiac muscle cell generates its own potential with these 4 phases just being described a little bit before. And depolarization goes along the membrane, and it opens calcium channels. Through these open calcium channels in the membrane, outer membrane of heart muscle cell, calcium from outside starts to go down the gradient into the cytoplasm of the cell. And here, this calcium triggers, stimulates opening of channels in the sarcoplasmic reticulum membrane. And through these open channels, calcium from reticulum goes and binds to troponin, which stimulates the beginning of the interaction of myosin and actin. And contraction begins, mechanical process of contraction. So you see, depolarization, electrical process, through calcium brings contraction. So calcium is intermediate stage, and this is coupling of the two processes, or simply excitation, contraction, coupling. So all this, let's consider again this sequence of events. First, heart muscle cell is depolarized due to conductive system impulses. This depolarization opens calcium channels in the cell membrane. Then calcium goes through these channels from extracellular fluid. Then these calcium ions initiate calcium release from reticulum, sarcoplasmic reticulum. Then calcium ions finally bind to actin filament, to troponin exactly, and initiate interaction of actin and myosin, which is contraction. So this is the full sequence of events in the excitation, contraction, coupling. Also, in this picture, we can observe how calcium is removed, because after each contraction caused by high calcium level in cytoplasm, the heart muscle, ventricles, and atria should be relaxed. For relaxation, calcium should be decreased again back to resting level. Otherwise, relaxation cannot be achieved. So it's important to know how calcium is removed. And actually it's easy. If it comes from two sources, it also is removed to both directions. First, of course, it's into sarcoplasmic reticulum back. As sarcoplasmic reticulum always contains much more calcium that we can observe in cytoplasm, the movement of calcium against the gradient can be achieved only by pump that uses ATP, only active transport. And calcium can also be released out from the cell by calcium-sodium exchanger. Now we observe how calcium goes to reticulum, and now how calcium goes outside through calcium-sodium exchanger. As you know, exchangers do not use energy directly. You see, here there is exchange of calcium for sodium. And driving element here is sodium that goes into the cell down its concentration gradient. But you easily may see that sodium entry without any help from regular pump will not be lasting for a long time, because sodium will be quickly accumulated in the cell and gradient will be lost and sodium will not enter anymore, so calcium will not be released. But sodium constantly is pumped out by usual pump, sodium-potassium pump that uses ATP, that is shown to the right side. So practically calcium-sodium exchanger works due to energy that is spent for functioning of the sodium-potassium pump. Now, the main law of the heart. We'll come back to this law many times, but right now you should understand it clear and try to memorize it. This law has two names, Frank and Starling. Both Frank and Starling independently did many works which became the basis for formulation of the Frank-Starling law. Actually, they didn't produce this full definition of law, but this law is strongly based on their experiments. They worked independently, and both names are used for naming this law. Otto Frank was the German scientist. You see, their works for basis for this law were done approximately in the end of 19th century and in the beginning of 20th century. They were almost born in the same year, 1 year difference. And Annulus Starling worked in London. He's an English physiologist. By the way, Starling should be a familiar name for you, because in the microcirculation, his equation was considered the Starling equation. He developed this equation to describe the processes that take place in the microcirculation system. All four pressures differences between hydrostatic and oncotic pressures of the two fluids that taken together provide balance between filtration and reabsorption from capillaries into capillaries in the microcirculatory system. This is also works of Ernest Starling. Now let's come to the law itself. It's rather easy, and actually it's based on the skeletal muscle, similar tendency. The basis is a length-tension relationship, and it's something that we already studied before in the muscles. And definition of the law can be done in many ways. Let's start from the simplest one. The greater the heart muscle is stretched during filling, so that greater length of the fibers, the greater is the force of contraction or tension. So the length-tension relationship, that's what the experiments of the both Frank and Starling were related to. So force of contraction and greater the stroke volume pumped into aorta. It's clear. Higher stretching, higher force of contraction, and more blood is pumped out of ventricle. But stretching occurs by blood that fills the heart. So blood that comes back from veins, that amount of blood provides stretching. So the venous return practically determines the force of contraction. And of course, this dependence is not endless. Until it reaches the maximum, it works. And maximum is achieved approximately at the level of 50% stretching, increase in length. In some sources, they are more cautious, they say 45%. But 45 and 50, for studying purposes, I think you can simplify it to 50%. So if initial length of fiber is taken for 1, so 1.5 stretching reaches maximal and maybe already starts to decrease slightly the force of contraction. So approximately about this level we can reach optimal, best stretching that contracts, produces strongest contraction, and then further stretching decreases force of contraction. But normally the heart operates within these limits. It's not very usual to stretch heart more than 50%, and so mostly that goes as a straight dependence, direct, proportionally, higher stretching by filling and higher force of following contraction. So the basis, now let's come back to the mechanism. But it's easy mechanism, this is the same that was described for skeletal muscle. Skeletal muscle entirely not stretched. You see? They say that in the middle of each sarcomere, the actin filaments already overlap, not simply meet each other, but already overlap. But at least they have the piece that goes closer to the center, and this piece will not be used by heads of myosin filament, and the distance till the end, to that line, is very short, so opportunity to contract is not great. So unstretched muscle produces very weak contraction. Here you see the moderately stretched muscle. The word moderately here means that it within this 50%. And actually this picture shows the best stretching, after which we already will not reach stronger contraction. But this exactly gives maximal contraction, because look, the ends of actin filaments are located just in the area where first heads of myosin appear. So all heads of myosin will participate in contraction, all of them, 100%. But there is no any unused portion of the actin filament. And this stretching makes the maximal distance for this optimal stretching, gives maximal distance from ends of myosin till that lines. So contraction may become stronger. And this really is the best way to stretch the muscle, optimal. Stretching, maximum binding of cross bridges, and maximal opportunity to produce strong contraction, which is muscle tension. And overstretching, more than 50%, produces very extremely weak contraction. Look here, this is excessive stretching of the muscle, and notice why it produces very little tension. Very small overlapping of filaments. Here, just a few heads of myosin can become attached to actin and try to pull them to the central sarcomere. But compared to the full amount of the heads of cross bridges, and this little amount that can participate, they cannot produce normal force of contraction. And exactly this situation is much more possible for the heart than to the muscle. Stretching like this we can achieve here in laboratory conditions, but normally the ends of the skeletal muscle are attached to the bones, and bones cannot be so much stretched cannot become so distant from each other. So such a great overstretching of the skeletal muscle is hardly achieved. But for the heart, there is no any bone attachment, and muscle can be stretched and stretched more and more depending on the amount of blood that fills the heart. And for example, if the heart is very weakly contracted, so the remaining volume of blood becomes high, an additional volume that constantly comes from veins greatly stretches the muscle, and it can produce very weak contraction as a result, because dilation of the heart greatly decreases the force. Great dilation, of course, overstretch. And this is a figure that shows the initial portion of the figure is exactly straight line, and diastolic volume that fills the heart increases, and stroke volume, as the index that shows how strongly heart contracts, is also increased proportionally. So it's linear dependence within normal reasonable limits. Further overstretching produces decrease of tension developed by the heart. But now let's recall main values for the heart. Stroke volume that serves as the index of strength of contraction. Absolute index is stroke volume, which is 70 milliliters on average and may vary from 55 to 90 milliliters normally. Then endostolic volume, we didn't mention it before. Ventricles become filled with blood from 90 up to 140 milliliters, and average value usually is taken for 130 milliliters. So when ventricles contract, they do not squeeze everything until the last drop. No, they release only 70 milliliters, and you can calculate that about 60 milliliters remain in the ventricle, which is called end systolic volume, by the way, this difference. And very important index used in clinical practice, this is ejection fraction. The word fraction itself implies that it should be part, and really this is a part made by stroke volume from the total end diastolic volume. How many percent of the volume are ejected? And if you divide 70 by 130, it will be slightly over 50. And really normal values of ejection fraction average make 55-60%, and limits range of norm starts from 50 to 75. Don't be scared, these values we are going to study next time when we are going to consider the cardiac cycle, and we'll come back to this again deeper, but right now if you have an idea about it, it's good. 50-75, this is a range of variations of healthy person resting ejection fraction. It's important resting because in case of physical activity and high work of the heart, this percentage increases. Stroke volume increase and diastolic volume increases, but it's not proportional increase. Stroke volume increases more, so that it makes higher percentage or higher ejection fraction. Look, classical 130 and diastolic volume corresponds to classical 70 milliliters. So, and now few more words about the two types of load for the heart. They are called the preload and afterload. Preload is the load by volume. Actually, end diastolic volume. Why it's called preload? Even before heart starts to contract, we already can see this load. It's already known that end diastolic volume stretches the heart, and if it's higher, it produces stronger stretching, and we can expect stronger contraction. It gives higher load for the heart, but the heart will respond by stronger contraction. Preload, so it's a volume, and diastolic volume, full volume accumulated in the heart before beginning of contraction. Note that simply diastolic volume will not be correct because diastole takes time, and during diastole, volume of blood in ventricles gradually increases and increases. So it's not exact value, particular value by end, but end diastolic volume is exactly volume that is reached by the end of diastole, and this volume stretches the heart and contributes to the stronger contraction during systole. So preload. And afterload is the load against which the muscle should produce strong contraction. Afterload for the left ventricle is the pressure in the aorta. And if you remember, pressure is determined by resistance, and sometimes we can say also about resistance. But pressure is simpler. So pressure is the afterload. Really, when ventricle starts to contract, at this moment valves are closed still, and pressure in the aorta is a diastolic pressure, and we should overcome this pressure and create a difference of pressures in order to produce ejection of certain volume, about 70 milliliters, for example, into aorta. So pressure is an afterload. And before contraction begins, this resistance and pressure is not develops. So it appears only when contraction begins. So afterload. Pressure by load by pressure. So it's easy. Load by volume is a preload. Load by pressure is afterload. Both loads increase, stimulate the stronger heart contraction. But as we already considered the influence of the stretching of the heart by end diastolic volume, it contributes itself to the further stronger contraction. So preload is easier for the heart to overcome. High overload also increases initially the force of heart contraction, so that heart can work against high pressure, but it cannot maintain for a long time so strong and strong contraction. So if afterload is chronically high, the heart gradually develops inability to overcome it, and at first, of course, it increases and increases, but very soon it can be not achieved anymore because the heart becomes weaker due to great afterload. But this is pathological physiology. Let's move to normal physiology. And this is the end of the lecture. Thank you for attention. Sorry if it takes a long time, but it's necessary and important portion of physiology. So let's stop.